Hi, welcome to Nagesh Biology online classes. Those who have not subscribed to my channel, please subscribe, like and share to your friends. Now we discuss about detailed entire information about Krebs cycle pathway. Krebs cycle, otherwise known as, there are different names are there, TCA cycle or citric acid cycle. Like this, there are different names are there. Total Krebs cycle events occurs in matrix of the mitochondrion. What are the major events occurs in the Krebs cycle? That we'll discuss. First, OAA. That is the full form of OAA, oxaloacetic acid. This is a 4-carbon compound. That oxaloacetic acid already it will be in the matrix of the mitochondrion. That OAA and acetyl-CoA. Acetyl-CoA, it is 2-carbon compound. OAA 4-carbon compound. This 2-carbon and 4-carbon compound both condenses with each other, leads to formation of citric acid. It is the first step. Citric acid 6-carbon compound. That is formed because of these two chemical compounds. Acetyl-CoA condenses with OAA, oxaloacetic acid. It is 2-carbon compound, this is 4-carbon compound. 2 plus 4, that leads to formation of 6-carbon compound, that is citric acid. First formed compound citric acid. So this pathway otherwise known as citric acid cycle. That citric acid consists of 3 carboxylic groups in its structure. 3 carboxylic groups. So this pathway, tricarboxylic acid cycle, because of formation of 3 carboxylic groups compound, that is citric acid. In the first step occurs presence of water. But this reaction, the total occurs in matrix of the mitochondrion only. All 10 steps, whatever I am going to explain, all 10 steps occurs in matrix of the mitochondrion. This step, that is called condensation. Here one coenzyme A, it comes outside because acetyl-CoA is there. So that CoA comes outside. Acetyl and OAA, that condenses, leads to formation of citric acid. That is the first step. This step occurs presence of citric synthetase enzyme or citrate synthetase enzyme, because citric acid is formed presence of that enzyme only. Citric synthetase enzyme that helps to formation of citric acid. That is the first step of the citric acid cycle. Second step, dehydration occurs. Removal of water. Removal of water, that step, that is the second step. That step name is known as dehydration. Removal of water from the substrate, that is called dehydration. That is the second step that occurs in citric acid cycle. Because of dehydration, that leads to formation of one chemical, cis aconitic acid. Once you observe, citric acid 6-carbon compound, cis aconitic acid also 6-carbon compound. Because of dehydration second step, that leads to formation of one chemical, cis aconitic acid. This second step reaction that is dehydration. That step catalyzed by one enzyme that is called aconitase. Whatever enzyme participates here, whatever enzyme involves, that is aconitase enzyme. Because of dehydration leads to formation of, that is also 6 carbon compound, cis aconitic acid. Next, that is the third step, that is hydration. Because of adding of water, that step is known as hydration. Addition of water molecule, that is called hydration. That is the third step. Because of hydration, one chemical is formed, that is citric acid. That compound also 6 carbon compound. Because of adding of water, that step number third step. Now fourth step, that is leads to formation of oxalosuccinic acid. That is also 6 carbon compound. But a major step, oxidation process, that occurs at a fourth step. That oxidation step, once you observe here NAD plus, that is reduced to NADH H plus. NAD plus reduced to NADH H plus, that is the energy form. This compound is reduced, but isocitric acid is oxidized. So that step, oxidation step, fourth step, oxidation. This third step, fourth step. Because of third step 6 carbon compound is formed. Because of fourth step also 6 carbon compound is formed. Number of carbons not changed here from here to here. 6 carbon compound, 6 carbon compound. Next here 6 carbon compound. After 4 steps 6 carbon compound only formed. But here that enzyme, once you see here third step, that is catalyzed by aconitase enzyme. Second and third steps catalyzed by same enzyme, that is aconitase. Whereas fourth step, that is the first oxidation in Krebs cycle. In Krebs cycle, that is the first oxidation. That oxidation step, this one, catalyzed by isocitric dehydrogenase. So name of the substrate based only, easy to remember. Isocitric dehydrogenase. That enzyme helps to conversion of isocitric acid to oxalosuccinic acid. But number of carbons not changed. So no change from here to here. After fourth step oxalosuccinic acid is formed. That also consists 6 carbon compound. Next, fifth step. Fifth step, decarboxylation step. Whatever the step occurs at fifth, that leads to formation of 1 carbon dioxide molecule for 1 acetyl CoA. For 1 acetyl CoA, 1 carbon dioxide molecule is removed. A removal of carbon dioxide from the substrate, that is called decarboxylation. 1 carbon removed in the form of carbon dioxide. This leads to formation of 1 compound, that is alpha keto glutaric acid. This is 5 carbon compound. 1 carbon, that is reduced from oxalosuccinic acid to alpha ketoglutaric acid. So oxalosuccinic acid has 6 carbons, but whereas alpha ketoglutaric acid, it has 5 carbon only, 5 carbon compound. 5 carbon compound, only 1 5 carbon compound is there in the citric acid cycle, that is alpha ketoglutaric acid. Fifth step, that is decarboxylation step. This step that is catalyzed by oxalosuccinic decarboxylase. It's a examination point of view, it is easy to remember. Oxalosuccinic decarboxylase, that is the enzyme involved in fifth step. Next, sixth step, two major events occurs. This is sixth step, decarboxylation and oxidation. Both the steps are involved. Once you see NAD plus, that is reduced to NADH H plus. Main step, that is oxidation. One NAD plus reduced to NADH. Next, here one more event also occurs. One more carbon removed in the form of carbon dioxide. So this sixth step, oxidative decarboxylation, because oxidation occurs, because of oxidation NADH is produced, and at the same time here decarboxylation also occurs. One more carbon removed in the form of carbon dioxide. Once you observe, fifth step, one carbon removed in the form of carbon dioxide. After sixth step, one more carbon removed in the form of carbon dioxide. At the end of the sixth step, one compound is formed, that is succinyl CoA. Succinyl coenzyme will form. But here the sixth step, one coenzyme A, that is involved in the reaction for formation of succinyl CoA. So once you observe, it has 5 carbons. Alpha ketoglutaric acid, it consists of 5 carbons. But whereas one carbon removed in the form of carbon dioxide, you can guess that how many carbons will be there? That consists only 4 carbon compound, that is succinyl coenzyme A is formed. That from here remaining all compounds up to regeneration of OAA up to oxalic acid, remaining all chemicals consist 4 carbons. Only one chemical that has 5 carbons in total entire citric acid cycle, that is alpha ketoglutaric acid. The sixth step, oxidative decarboxylation, this step catalyzed by one enzyme, that is alpha ketoglutaric dehydrogenase. Wherever oxidation occurs, that enzyme ends with dehydrogenase. That how to remember? The enzyme name alpha ketoglutaric, oxidation step dehydrogenase. After end of the sixth step, succinyl CoA is formed. Next, seventh step, that is the major step, cleavage step. So because of cleavage, succinyl CoA, it undergoes cleavage that leads to formation of here coenzyme A, that is released. But the major event whatever occurs in this step, that is the seventh step, ADP plus inorganic phosphate leads to formation of ATP. But one thing you can observe, here this reaction occurs presence of water, but it is water is not involved. This reaction also occurs presence of water, but water it is not involved in the reaction, but the total reaction occurs presence of water. Succinyl CoA 4 carbon compound that leads to formation of succinic acid because of cleavage step. So whatever carbons are there in the succinyl CoA, same number of carbons also found in succinic acid. Once you observe, succinyl CoA has 4 carbons, but here the succinic acid also consists 4 carbons. But the seventh step, That is the most important step for formation of one single ATP molecule. But whereas in the animal cells, animals, GTP is produced at the same step. That is the guanosine triphosphate. That guanosine triphosphate, that is equal to one single ATP molecule. This reaction cleavage step, that reaction occurs presence of thiokinase, that enzyme catalyze to form succinyl-CoA to succinic acid. Till now what we discussed, total 7 steps of the Krebs cycle we discussed. Now 8th step, once you will see here, this is 8th step. Because of 8th step, here fumaric acid is formed. This fumaric acid, it also consists 4 carbon. But here the 8th step, one major event occurs. FAD plus, that is reduced to FADH2. 8th step, that is oxidation step. Only 8th step, only FADH2 produced. Total 4 oxidations occurs in Krebs cycle. At 4th step oxidation, 6th step oxidation, 8th step oxidation, and 10th step also oxidation occurs. Out of 4 oxidation reactions, only 8th step, that is at the time of conversion of succinic acid to fumaric acid, because of oxidation 3, FADH2 is produced. So only FADH2 produced at 8th step, that is oxidation 3. But enzyme succinic dehydrogenase, succinic acid is there, the succinic dehydrogenase enzyme involves for formation of succinic acid to fumaric acid. At that time, one single FADH2 produced for one acetyl-CoA molecule. Next, 9th step, fumaric acid converts into malic acid is formed. This is the 9th step. But at 9th step, here the 3rd step, water is involved. Here also at 9th step also, one single water molecule is involved. So whenever water molecule is involved in the reaction, that step we can call it as hydration step. Here 3rd step is hydration occurred. Next 9th step also hydration. Because of hydration, what happens? It leads to formation of malic acid. That malic acid, it consists of 4 carbon. Because from here onwards, succinyl-CoA, succinic acid, fumaric acid, malic acid, all these they consist 4 carbons including OAA. That is the first step, condensation step, participated one chemical, that is involved a chemical, oxaloacetic acid. So that is 9th step, because of involvement of water, hydration step, malic acid is formed. That enzyme here, that one enzyme is involved, that is fumarase is one enzyme is involved. That enzyme helps to conversion of fumaric acid to malic acid. Total what we discussed earlier, total 10 steps occurs. Its last step, that is the 10th step. But because of 10th step, that is oxidation 4. Because of oxidation 4, once you see here, NAD plus reduced to NADH, H plus is produced in 10th step. Here once you observe, at 4th step oxidation, 6th step oxidation, 8th step oxidation, and as well as 10th step also oxidation. Total and 4 steps oxidation occurs. At 10th step, one more NADH, H plus is produced. That is last step, 10th step. That 10th step name oxidation that is the fourth oxidation. This step that is catalyzed by because of one particular enzyme, that is easy to remember, malic acid is formed at the end of the ninth step. So enzyme name malic dehydrogenase. That enzyme helps to conversion of malic acid to oxaloacetic acid, that is regenerated OAA. That OAA again enters into the pathway. Like this, total 10 reactions occurs in the Krebs cycle. All these reactions discovered by Sir Hans Krebs, scientist. Because of discovery all these reactions by the Krebs scientist, so he also received Nobel Prize. All these reactions occurs in matrix of the mitochondria. But all the steps, the major steps names are, that is examination point of view, that is very important for steps names. So I'll write here steps name. First step, this is condensation. Whatever step occurs here, that is condensation. Removal of water, that is called dehydration. So removal of water, that is dehydration. Here water is involved. Third step. So that reaction is called hydration. Total 3 steps. Finish. Now fourth step, that is oxidation. First oxidation in Krebs cycle. Now I am saying that in Krebs cycle it is first oxidation. In Krebs cycle it is first oxidation. So fifth step, carbon dioxide is removed. That is called decarboxylation step. First decarboxylation of Krebs cycle. Next, sixth step, oxidative decarboxylation. Because here oxidation occurs and decarboxylation occurs. So oxidation, that is second oxidation. Decarboxylation also second one. Second oxidation of the Krebs cycle and the second decarboxylation of Krebs cycle. Next, seventh step is known as cleavage, because of splitting of this compound. So that is otherwise known as cleavage step. Eighth step here, FADH is produced. That is oxidation only. Like fourth and sixth step, whatever so compounds are formed here, like that compound is formed, but FADH is produced. Because of so formation of eighth step FADH2, that is produced. This is also, we can consider this is oxidation, because it is reduced, it is oxidized. It is third oxidation. Third oxidation of Krebs cycle. Ninth step, water is involved. Third step, ninth step, the major steps, that is hydration steps, because of involvement of water. So the ninth step, water is involved. That's why that step is known as hydration. Last step, that is the 10th step, that is oxidation. 4. Total 4 oxidations, 2 decarboxylations occurs in the Krebs cycle. All these are 10 major steps of the Krebs cycle, catalyzed by 10 different enzymes. From this pathway, what type of questions we can expect in the exam? For 1-acetyl-CoA enters into the Krebs cycle or citric acid cycle, how many CO2 molecules are produced? 2 CO2. For 2-acetyl-CoA, how many CO2 molecules are produced? 4 CO2 molecules. Like that, here different types of questions you can expect. At 7th step, that is a very most important step. In animals, at this step, in animals, at 7th step, GTP is produced, guanosine triphosphate. That guanosine triphosphate, it helps to formation of one single ATP. In animals, GTP is produced at 7th step, but whereas ATP produced in the plants, that is the substrate level phosphorylation. That is the first substrate level phosphorylation of Krebs cycle. This total